It is a pleasure to present this research work from the Department of Cancer Chemoprevention, Chittaranjan National Cancer Institute, Kolkata. This work has been carried out under the guidance of Dr. Subhadip Hajra. The study aims to develop a smarter treatment strategy for triple negative breast cancer by combining a natural anti-cancer compound with chemotherapy using an advanced drug delivery system. To appreciate why such an approach is needed, let us first understand the challenges associated with triple negative breast cancer. Triple negative breast cancer, or TNBC, is one of the biggest challenges in breast cancer treatment, particularly in India. Breast cancer is now the most common cancer among Indian women, and nearly 1 in 3 patients may have triple negative subtype. Compared with Western countries, TNBC tend to affect women at a relatively younger age, grows rapidly, spreads early, and has limited targeted treatment options. These challenges highlight the urgent need for more effective treatment strategies. TNBC is particularly difficult to treat because of a small population of cancer stem cells, often referred to as the roots of the tumor. These cells can survive chemotherapy and undergo a process called epithelial to mesenchymal transition, or EMT. This makes them more mobile and invasive. As a result, they spread to other organs, form new tumors, and can cause the cancer to return even after treatment. This is why TNBC grows aggressively, spreads early, and remains one of the most challenging forms of the breast cancer to treat. Modern treatment of triple negative breast cancer usually involves a combination of 5 main pillars: surgery, which physically removes the tumor; chemotherapy, which rapidly kills the dividing cancer cells; immunotherapy, that helps the patient's own immune system recognize and attack the cancer cells; targeted therapy; and the fifth pillar, which is the focus of our research, is smart drug delivery. The study focuses on advanced drug delivery using nanoparticles and exosomes to deliver drugs more precisely to tumors, improving treatment effectiveness while reducing side effects and drug resistance. Chemotherapy remains the mainstay of treatment for TNBC. There are several classes of chemotherapy drugs called anthracyclines, taxanes, platinum agents, alkylating agents, and others. Among these, doxorubicin is one of the most important and widely used drugs. It works by entering the cancer cell's DNA and stopping it from multiplying, leading to cancer cell death. However, a major limitation is that a small group of cancer stem cells can survive this treatment, eventually leading to drug resistance, cancer recurrence, and metastasis. This highlighted the need for a more effective therapeutic strategy. We need better strategies, like combining it with other agents and using smart delivery systems to overcome this resistance. Another major limitation of doxorubicin is that it can cause side effects, and over time, they may enrich cancer stem cells and promote EMT, the process that makes cancer cells more invasive and resistant to treatment. As shown in the graph, doxorubicin increases cancer stem cell markers as well as proteins associated with EMT. To overcome this challenge, the study combined doxorubicin with 3,3-diindolylmethane, or DIM, which is a natural compound found in cruciferous vegetables such as broccoli and cabbage. Together, this DIM and doxorubicin helps suppress cancer stem cell enrichment and inhibit EMT, suggesting a more effective strategy than chemotherapy alone. Our results showed that when we treated cancer cells with a combination of DIM and doxorubicin, bad markers like encadherin went down with simultaneous upregulation of good markers like encadherin. It also reduced important EMT transcription factors such as snail, slug, and vimentin. Most importantly, the combination dramatically increased apoptosis, the programmed cell death in the cancer cells. At the same time, the combination showed less toxicity to normal breast cells, which are very encouraging. Overall, the results that we got demonstrated that the combination of DIM and doxorubicin worked better together than doxorubicin alone. Next, we developed an exosome-based drug delivery system. First, both DIM and doxorubicin were co-loaded into mesosporous silica nanoparticles. These drug-loaded nanoparticles were then encapsulated within the natural exosomes, which acted as biological carrier with excellent biocompatibility and tumor targeting ability. Once delivered to the TNBC cancer cells, the drugs were released inside the tumor cells, leading to inhibition of EMT. We successfully developed our drug-loaded nanoparticles and confirmed that they could carry and gradually release both medicines. More importantly, they reduced the changes that helped cancer cells spread and suppress cancer stem cell growth. One of the key aspects of the study was the use of exosomes as the natural drug delivery vehicles. Exosomes are tiny vehicles that can circulate longer in the body, naturally target tumors, and protect the drugs that they carry, making them highly effective carriers for targeted drug delivery. This slide explains comprehensively about why we combined our nanoparticles with exosomes. Nanoparticles are powerful drug carriers. They are excellent at carrying large amounts of drugs and releasing them in controlled manner. They can be quickly cleared by the immune system and have limited penetration into the tumors. Exosomes naturally overcome these challenges. They circulate longer in the body, evade immune system, and have an inherent ability to home to the tumors. By encapsulating our drug-loaded nanoparticles inside exosomes, we combine the strengths of both systems to create a smarter drug delivery platform with improved targeting, deeper tumor penetration, and enhanced therapeutic efficacy. Finally, in the cell culture experiments, our dual drug-loaded exosome nanoparticles effectively reverse EMT. They decreased encadherin slug and vimentin protein levels while increasing the epithelial encadherin. This shows the treatment is shifting the cancer cells back to a less aggressive state. In the animal models using patient-like tumors, the dual treatment shows excellent results with significantly reduced tumor volume and burden. Animals treated with dual exosome burden had better survival rates. To conclude, the study presented a smart and targeted approach for treating TNBC by combining a natural compound DIL with the chemotherapeutic drug doxorubicin using an exosome-based drug delivery system. The strategy improved targeted drug delivery, reduced tumor growth, enhanced cancer cell death, and helped overcome treatment resistance in preclinical studies. While further clinical validation is needed, these findings highlight the promises of the advanced drug delivery systems in developing safer and more effective therapies for patients with TNBC. The findings of this study have been published in Journal of Nanobiotechnology, highlighting the potential of exosome mediating co-delivery of DIM and doxorubicin as a promising targeted therapeutic strategy. Finally, I would like to sincerely acknowledge the support of ANRF, formerly served, for funding this research. I also thank Dr. Jayanto Chakraborty, Director CNCI, Dr. Prosenjit Shaha, Head of the department, and all the members of Cancer Chemo Prevention Laboratory for their guidance, support, and valuable contributions.